Welcome to Study Loop.
In order to answer this question, we have to discuss two separate but interconnected concepts. So the first is our nomenclature with regards to what all these different strands are. So if we are to look at our DNA strand, of course, it's going to run in the 5' to 3' direction. And complementary to this, we can call this coding strand. Complementary to this coding strand, we have another strand that is anti-parallel, running in the 5' to 3' direction, but it will be this way in the 5' to 3', whereas our coding strand was to the right in the 5' to 3' direction. And we can call this anti-parallel complementary strand the template strand. Now, when we are going to produce an mRNA transcript, right, we are going to use our coding strand. So we take the same coding strand right here, and we're going to move it over 5' to 3', and we are going to produce a 5' to 3' RNA product. So this was our DNA coding strand, and this is our RNA that we have now transcribed from the DNA. But as you can see, the orientation is the same as our original DNA template. So in both cases here, these were both DNA. So what has changed? What is the difference between this template DNA and the RNA that we transcribe? Well, the difference is that we are going to have thymines in our original template DNA, whereas in our RNA, we are going to have uracil. And this brings us to our second point that we need to discuss. What is the difference between thymine and uracil? Well, if we look at the structure for thymine, we find that is actually very close to the structure for uracil. And actually, perhaps let's start with the structure for uracil, just because you'll see in a moment why we're doing it this way. So both, of course, are going to be these cyclic structures. So if we just draw them out, we're starting with our uracil. There's a double bond right there. Okay, we have a double bonded oxygen over here. We've got a nitrogen right there. It goes off to form a methyl group right here. Another double bonded O, and we have another nitrogen right here. Now, of course, fill in the hydrogens where you would expect them in the ring. So you could actually say that this is an H right here, but we'll leave you to fill in the hydrogens. And of course, like we said, this is a methyl group right here. So this is a CH3. But you should be able you should be familiar with this kind of a structure that we draw. So now let's go over to thymine. Okay, so for our thymine, same thing, same kind of ring structure. We've got our nitrogen here. We've got our methyl group attached to it. Then going around the structure, we've got a double bonded O, and we've got an N right here, another double bonded O, and we are going to have a double bond here. So up to now, everything is the exact same between thymine and uracil. The only difference is thymine has an additional methyl group right here. So we can point out that this is a methyl group or a CH3. Okay. So what's the difference between thymine and uracil? Well, essentially, thymine is just methylated uracil. So going back to our first point, we said that these two strands are identical, right? Our template DNA and our RNA that has been transcribed from our coding DNA strand are exactly the same, save for the fact that thymine is present in our template DNA, whereas uracil is present in the RNA transcript. However, the only difference between uracil and thymine is that thymine is, it has this extra methyl group on this carbon right here, whereas this carbon is unmethylated. So in answering this question, we have to remember this concept that if we are to methylate uracil, or in other words, the methylation of uracil yields thymine. And of course, as we know, the template, once again, the DNA template and the RNA transcript are going to be exactly the same, except every single place on the DNA template where you saw a T, it will now have a U. So that means that they will never be the same exact sequence, because of course there's the difference between the T and U. But in the case that we find methylated uracil, we can actually produce thymine. So we can actually go from this uracil and through methylation produce thymine. And that's really the point that we're trying to drive home here, is that the methylation of uracil can produce thymine in certain circumstances. The uracil might get methylated. And this means that we actually will have identical strands, because now everything aligns. Not only are they running in the same direction, right, 5' to 3', right to left, but now they will have A's, C's, T's, and G's in all the same spots. So that means that if we are to look at our final answer in terms of our answer choices, okay, the correct answer is answer choice C.